Okay, I've got a couple of handouts for you. One is something I had for you on Tuesday, and anyone in hybrid who didn't get this, they are online, but I have the PowerPoints for Chapter 20 on the lymphatic system. If for some reason you did not make a copy or want a copy, that's here for you. I also have a handout that will make more sense by the time we finish up here. This is, last exam I gave you that blood chart as a preview of what will be on the test. This time I'm giving you a little spirometry chart. This is the kind of measurements we'll be doing involving the lung. We'll be doing these activities next week in lab, but you'll actually need to be able to do this for the exam as well. So let me pass these out, and if you would just pass those around, Tony, if you don't mind. And I will post this on Blackboard as well for those who aren't here. I'll also have copies with me next Tuesday in lab. Also receiving the case studies. So those grades will be in there by tonight. The second case study, the second group of case studies will be due on Thursday, July 30. Okay, Thursday, July, that's the last day of regular classes. So Thursday, July 30, that's the week of our urine lab. It's the last full week of lecture, and that is the day that the second case studies will be due. Same idea, hard copy, signed by your group, and uploaded, one person upload to Safe Assign. You can have that done anytime, right? But no later than Thursday, the 30th of July. The only reason that groups are losing any points on their case studies is either a lack of or incomplete citations. So just make sure you have citations. You know what they look like. They have an author, a name, a year, a journal, whatever, and make sure that those are there and complete. www.webmd.com is not a citation, right? Never has been, never will be. So that is not an acceptable citation for credit. So make sure you have proper citations and you've done the best you can answering the questions, and you will receive full credit. Fair enough? Okay, so let's take a look. We've got an exam next Thursday. It'll be a full-length exam. It'll cover lymphatic. It will cover digestive, and it will cover respiratory. I almost finished digestive with you last time. We're at the very end talking a little bit about the liver and the pancreas, and then we'll be diving into the respiratory system. I will not finish respiratory today. I will finish it on Tuesday, and likely we'll have some time at the end of that, as I like to do, to answer any questions that you may have. That means that you should try to come next Tuesday as prepared as possible, having done lymphatic quiz, having done digestive quiz, with any questions you might have about respiratory, knowing we haven't finished it, but anything at all that you're struggling with, bring with you that day so I can address it. Then the final exam, remember that the lab exam will be on Tuesday. I'm asking the exam to be at one of two times. You'll either come 4 to 6 or 6 to 8 on Tuesday, the 4th of August. And that would be, there won't be a lecture that day. So I don't want you to have a lecture in a lab and all that on the same day. So it'll be 4 to 6 or 6 to 8 for the lab exam on Tuesday. And then on Thursday, we're not going to have a class exam. And I don't think I made this announcement Tuesday lunchtime lab. The final exam will be in the testing center for everybody, and I'll give you flexibility as to when you take it, so that now you have a little bit more flexibility. You may have other classes, and I'll allow you to take it on Thursday or Friday of that week. So Thursday or Friday, more about that coming up, but you don't necessarily have to be here at 4 o'clock on that Thursday, and if for some reason you want to take it on Wednesday, I'll allow that. Okay, if you want to get out of town earlier or whatever, but I want to give you maximum flexibility, knowing we have 2 exams that week, one lab exam and one lecture exam. That lecture exam will be partially cumulative, not hugely, probably 20% of it or so will be cumulative, and I'll give you more notes about that portion of the exam. It won't be the minutia, it'll be more global, bigger ideas of how one system communicates with other systems, but it will be worth a little bit more points. Rather than being worth 80 points, rather than being worth 90 points, it'll be worth 120. So your final exam is worth a little bit more because of the cumulative nature of it. I think that kind of gets me through all the details. Anything that's you're wondering about, we've only got 2.5 more weeks of class. Really 3 weeks from today, right? 3 weeks from today, you'll be done. It'll be the final exam, basically. So we're coming in rapidly toward the end. I want to make sure you've got all your calendars figured out and you know what to expect. Remember, too, that there will be, by the time we're done with this, a total of 12 mastering lecture quizzes. I will drop the lowest 2. In addition to those 12, there are 2 extra credit quizzes. The problem is, no problem, but every 30 minutes when Mastering and Blackboard resynchronize, it puts those extra credit quizzes back to being worth 10 points. They will be only worth 0. So any points you earn on those extra credit quizzes will be extra credit points. But just keep in mind, even if I keep changing the darn thing that the quiz is worth 0, every time it updates, it says, oh, it's worth 10 points. So it will reflect that, and you may see your grade moving back and forth as it does that update, okay? But other than that, those will be extra points. I will also drop the lowest lab quiz grade, and you've got your mastering points coming, you have your 2 case studies, you have your lab report. So lots of points coming your way still. Any other concerns? Logistical things. Chelsea?
July 30th, the male reproduction will be online, and then the female.
Yes, I don't recall exactly, but yes, the 30th will be the last, quote, lecture, and there will be a part of the reproductive system online as well. Like you're doing lymphatic this time online, the reproductive will be online. And the reproductive, quite honestly, is, again, mostly a review. There's not much new material. So that will be, I think, of the 3 chapters on the final exam, the reproductive is the kinder, I think, least struggling. The urinary system is a little bit more of a monster when it comes to the physiology of that, and that's why I would definitely want to do that live with you and leave the reproductive materials to online. So there is a reason why I choose what I do, but it may not always be clear. Let's take a look at the digestive system at the end. Actually, what was probably the brand new thing, the thing that is least familiar to you, would be the 5 hormones. So what were those 5 hormones? The 5 hormones, 4 of which were made in the duodenum, 1 of which was made in the stomach, and I am probably all in the wrong place. Give me a page on that. Am I getting close?
Yeah. 2214.
2214. Okay, thank you. So what were those hormones, folks? The very first one, just as a quick review, gastrin, made by the stomach, specifically by the G cells, right? And this did what? This hormone was stimulated by the very presence of food in the stomach. So once you eat, kerplunk, food's in the stomach, and now this will turn on gastrin, specifically high protein content. This is going to cause your stomach to start making more acids and enzymes and increase the mobility of the stomach. Then you've got secretin. Secretin and the rest of these are all made by the duodenum. Secretin, once you have food or chyme now dropping into the duodenum, that's going to pick up secretin. Secretin is now going to increase the release of bile from the liver and the gallbladder and the buffering capacity of the pancreas, and, as we said before, slow down motility so that the digestive system now has more time to absorb those nutrients that have now arrived into the small intestine. You've also got GIP, the gastric inhibitory peptide. This one's also going to be released especially when fats and sugar enter into the small intestine. What was the other one that did fats? Not only was GIP important for fats, but also the next one, CCK, right? So when either one of those drop into the duodenum, when fats drop into the duodenum, you're going to upregulate GIP and you're going to upregulate CCK. And in addition, GIP is turned on by glucose, which also has an effect over in the pancreas to upregulate the beta cells to make more insulin. Okay? And then we get over to CCK. And CCK was also going to be picked up or secreted more when fats arrive in the duodenum and some proteins. This is going to increase your secretions from the pancreas and cause that squeezing of the gallbladder to help deal with those fats. And then finally, you've got VIP, vasoactive intestinal peptide, and this one is going to basically dilate the capillaries so that more nutrients can be absorbed into the bloodstream. And it'll also say, hey, stomach, we've had enough acid right now. So basically, if you think about this, you got these check and balance system. As things are moving through the digestive system, one thing has an effect downstream, and oftentimes it also has an inhibitory effect going back more proximally. Then, and tell me if there's anything at all along the way that I said last time that I need to restate. I'm heading down toward the liver. We skipped over teeth, and liver started on 22.21. So I want to pick up here, unless there's something specifically that you want me to address now, or anything from the mastering quiz on digestion. I think the lymphatic quiz is due like today, tomorrow, soon. So make sure, if you haven't jumped on lymphatic, that you've looked at that in anticipation of that quiz. The digestive quiz is late in the weekend, or Monday. It's coming up. And then the respiratory quiz will be the last and final quiz. I know that one's due Wednesday night, the night before the exam. So they're spaced out, and there's some homework assignments associated with them as well. Okay, so liver tissues. Let me just start all over again. When I say liver, what comes to mind? Just give me some popcorn ideas about what the liver does for us, for you, for me. Plasma proteins, right? So we think back to cardiovascular in the blood, definitely albumin and fibrinogen and all those proteins made and found in the blood are mostly made by the liver. We also learned in the blood chapter that liver and the spleen help to break down your red blood cells. They help to recycle heme. They're hugely important with that. We are appreciating, as we step into this unit, that the digestive system relies upon bile, right, that is released by the liver, stored by the gallbladder, and released when CCK and GIP, right, are activated. We're going to get more bile released. And right now, as we're in the digestive system, we also want to imagine that molecules that are absorbed into the gut, going back just a moment ago, which hormone caused greater dilation of the vessels to increase absorption? VIP, right? The vaso, right? Vaso. So that one's going to increase the dilation so that we can absorb more nutrients. And those nutrients are going to travel first to the liver. So as you absorb nutrients into the bloodstream, they're going to first go to the liver. And what vessel is carrying them to the liver? Hepatic portal vein, right? That is the vessel that brings all these newly absorbed nutrients to the liver. The liver, as we're going to see in a moment, is then going to, quote, filter through all that. And it's going to make sure that there's nothing in there that could potentially harm us. There are a lot of macrophages in the liver. Those macrophages could do what? Gobble up and destroy anything that those cells sense as being dangerous or problematic for you. And then that blood, with now the filtered nutrients, is going to continue out of the liver in what vessel? We got there through the hepatic portal vein. We're going to leave out of the liver and join the rest of the circulation through the hepatic vein. Right? So the portal word simply is referring to what comes in, what comes into port, right? What comes into port, that's coming into the liver. What leaves the liver, do you agree, is still deoxygenated, right? It hasn't suddenly gotten red, so it's still blue blood. It's simply been filtered, and that's going to jump back into the inferior vena cava, right? It's going to go from the liver to the inferior vena cava via the hepatic vein. Okay, so with that in mind, let's figure out what the liver looks like. It is made of 4 lobes. Do you remember what those were from last time? It's pretty easy. Anybody remember the 4 lobes? Right and left. Yep, it's getting easy, right? And then there was a caudate and a quadrate, right, that were more visible from the backside. So 4 lobes of the liver, but microscopically, we say that the liver is broken up into hundreds of thousands of lobules, right, small lobes. And each of these little lobules is about 1 millimeter in diameter and is separated by a septum. These lobules are basically a hexagonal shape as we look at them, and each of these hexagons is going to have on its corner, right, there's 6 corners, if you will, 6 edges, no, 6 points on hexagon, and each of those points has at it a hepatic triad. that triad is the coming together of three vessels. It's going to have hepatic portal vein. In other words, this is where the nutrients are coming in. Okay? There's also going to be some hepatic artery there and also some bile duct coming up. And those three are going to come in close proximity. So you're going to have a red, a blue, and a green, right? An artery, a vein, and a bile duct that'll be showing up as green. The lobules also contain a large number of hepatocytes. Those are the cells that make up the liver. And again, those are arranged in these plates that create these spoke-like structures. And these plates are very thin, 1 cell thick. And these plates have short microvilli on them to increase surface area. And these plates, 1 cell thick, also are separated by sinusoids. That is, very delicate blood vessels that we could describe as being either fenestrated or sinusoidal, right? Very large fenestrations, essentially sinusoidal or, quote, discontinuous capillaries. That makes what's going to happen here possible. So let's take a look at what I've just said to you and put this together. So we're down in the microscopic structures of the liver. About 100,000 of these, I'm just going to keep drawing on this, but there is the hexagon. That would be a lobule, okay? And what you see is that each lobule is separated by, and you see the lobule is broken up into these spokes. I'm just going to draw over this, right? You kind of get these spoke ideas. Kind of looks like a wheel, doesn't it? And what you've got now is those spokes are one layer of hepatocytes. And in between those spokes, do you see the spaces? Right? Do you see the so these are big spaces in between these cells. Those are the sinusoids. And those sinusoids have fenestrated capillaries. So holy capillaries where a lot of things can exchange. Then, this is new, in the very center of each lobule is what is going to go to the the hepatic vein. So watch my pointer as I walk you through this. So what that means is that blood is going to come in, nutrients are going to come in here at the triad as a portion of that hepatic portal vein. Those nutrients are going to be moving. See all these spaces? Those nutrients are going to be moving toward the center. And as they go toward the center, they're going to be picked up by the central. This is a branch. This is going to go off to the hepatic vein. So as these nutrients come into the liver, they're going to move toward the center of the lobule. Along the way, they're passing a whole bunch of hepatocytes. Those hepatocytes are surrounded by these big spaces where there's a lot of macrophages and other cells that can capture foreign objects. And then it's going through these sinusoidal spaces that the blood is going to be, quote, cleansed and detoxified, and we're going to be looking for things that could harm us. And then that blood gets picked up here. It kind of drops into this middle vein, and then that's going to head off to the hepatic vein. So it's going through all these little spoke areas. And imagine all the surface area here, right? This is a lot of surface area. But look what else is coming in here. What else is coming in here is not only the blood from the hepatic portal vein, but also some blood from your hepatic artery. What would the hepatic artery be sending? Nutrients, right? So we've got nutrients coming in, we've got cells coming in, and then there's all these big sinusoids, which mean that the blood, the venous blood and the arterial blood actually kind of gets to mix here. Okay, this is an unusual place. The liver kind of receives both arterial and venous blood, and it's kind of mixing. And that mixing allows for the purification of and the cleansing of the blood. So here we go. Here's the words. So blood is going to come in to the liver sinusoids from branches of the hepatic portal vein and the hepatic artery. About 1/3 of this blood supply that's going to come in is arterial. About 2/3 is venous, right, coming in from the hepatic portal vein. And here, all of this kind of gets to merge together in this big, vast party room. So all these cells and all this blood is going to get to mix. Those hepatocytes on either side of those spokes are going to help regulate what comes in and out. We know that the hepatocytes, remember, are the cells that make the albumins. They're the cells that make the liver, the blood cells, right? The blood proteins. So these are the cells, right, that make the albumin and the other cells. So they're the proteins. So these are all mixing together. And here also in the liver, I mentioned there were a lot of macrophages. Well, those macrophages were described by a guy named Kupfner. So these are the Kupfner cells, right? Just another name for the macrophages of the liver. You just got to know the guy's name. So these cells are gobbling up anything that's damaged. Remember we talked about how the liver breaks down red blood cells? How's that happening? Those red blood cells are coming in what vessel? They're coming in the artery, right? They're coming in the artery quite easily, aren't they? They're coming in the venous side too, but they're coming in, and they are, anything that's old and damaged, about 120 days old, is going to get chopped up by these Kupfner cells. Also there, any kind of bacteria or cell debris or anything that could possibly be there that the cell says is dangerous is going to be engulfed. Remember too that this is also where we absorb heavy metals. So should you have any heavy metals in your blood, right? Tin, mercury, those are picked up and stored in the liver. And iron. Remember too that liver is a place of iron storage. We talked about that a long time ago, as I recall. So all of these things are happening in the liver. And everything in that sinusoid, in that spoke area, those big party rooms, is then going to drain toward the central vein. The central vein was that vein in the center of each of the lobules. And then that will go back toward the hepatic vein, eventually drain back into the inferior vena cava. It's an amazing thing. Now, also, the hepatocytes are secreting bile, right? The liver makes bile. And that bile will be collected into those bile ducts. The triad, right, was three things. The green part was the bile duct. That's actually collecting the bile and sending it where? Eventually out to the duodenum, but also some of it's being stored in the gallbladder. So it's interesting. You've got this two-way direction going on, and let me show that to you in this cartoon. So, do you see where we are? This area here would be considered a triad. It's one of the corners of the hexagon lobule, and we see again the hepatic portal vein bringing blood in, and then watch. Can you kind of see this? If you kind of watch and imagine this blood's coming in, right, into this hepatic portal vein, and then it's going this way, isn't it? It's going in this way, and it's passing through these sinusoidal areas. Eventually, it's going to dump into that central vein. Along the way, it's going by all these hepatocytes with these Kupfner cells that are phagocytic. Also here, you've got blood coming in from hepatic artery, and I told you about 1/3 of the blood going through here is arterial, 2/3 of the blood or so is venous. And then, what was this dude? This was a branch of the of the bile duct. These same cells, right, these same hepatocytes back here are making bile. The bile is coming back out. So let me try to make that more clear. I think the liver can be a little bit confusing. This is the direction of blood, right, coming into the vein. This is the direction of the arterial blood. But the bile is being produced by these cells and going this way. So what you've got is this cross communication. You've got diffusion, if you will, of molecules moving, though, cross. This is a very busy area, right? This big party room where things are moving. So you've got bile being made and dropped into the bile duct. You've got blood being filtered as it passes through all these sinusoids. Phenomenal, phenomenal, complex. Can't live without it, right? We can't live without the liver. So that's why we need to save the liver. And the things that in our culture attack the liver and cause most need for liver transplants are hepatitis, different versions of viral infections that can be very, very damaging to the liver, itis, right, inflammation of damage to the hepatic, the liver, and also alcohol can be very damaging to the liver in excess. What happens is, in part of this, is in that disease state, the blood flow starts to get blocked up. So the blood can no longer move through the liver like it should. There's scar tissue and damage to the liver, and that starts to increase portal hypertension. Not hypertension to your entire body, right? Not to your entire vascular system, but to the liver itself, there's a portal hypertension. And as you begin to increase the pressure in the hepatic system, that is going to also start distending small veins and capillaries here, and that will lead to the rupturing of the liver. Well, if the liver can no longer work, then you're going to no longer, get rid of toxins, but also that can lead to very critical bleeding. Okay? And fluid backup can lead to ascites, and that's the word for that excessive fluid buildup that can happen in the gut. When the liver is in trouble, right, either through hepatitis or alcoholism, or any kind of damaged liver will typically have ascites as a side effect. And that is the liver, okay? It's a very quick trip, but I want you to appreciate sort of the big idea about the liver. The take-home, the homework, and a couple questions on the quiz will help you focus in on what's important. 2 more accessory organs in addition to the liver. We've got the gallbladder and the pancreas. Gallbladder, you've seen it. You know where it is. It's up underneath the liver in the fetal pigs and in you. If you look at it, it's up under the liver's right lobe. And the gallbladder itself can be divided up into 3 regions: the fundus, the dome, the majority of it, the body, and a narrow region called the neck. This is going to be storing and concentrating. So not only is it storing excess bile made by the liver, waiting for CCK to tell it to squirt, but it's also helping to concentrate it. So the bile is more concentrated here. Bile and the content of bile is important in emulsifying, breaking down fats. And if you don't have the fats emulsified, then they can't travel, right? We know this whole story. We know that fats can't travel through your blood, right? Because fats are hydrophobic and the blood itself is hydrophilic, and water and oil don't mix. So how do you basically get mayonnaise? How do you get fats to emulsify, to mix with watery things, right? And bile is what does that. So in your digestive system, the bile is emulsifying, breaking down the fats, making them so that they can, quote, dissolve in or at least be in association with the rest of your body. So how does this work? So you have from the liver, right? From the liver, there are the right and left hepatic ducts. Those 2 ducts, the right and left hepatic duct, carrying bile, newly made bile, are going to merge into what's called the common hepatic duct, right? Common usually suggests to us that something has come together or has separated. And then the bile will flow from the common hepatic duct from the common hepatic duct to the common bile duct. That's a direct route to the duodenum. And that would enter where? Do you remember where that drops into the duodenum? If you look at a digestive model, you'll see a little nipple-shaped structure where the bile drops into the duodenum, and that's sometimes called the hepatic pancreatic ampulla or the duodenal papilla. We'll see that in a minute. And there's also sort of a flowing backwards of this bile in the cystic duct. Okay, so when there's extra bile, it kind of backward flows back into the gallbladder for storage. And then here's those words. I apologize, one step ahead. So the bile duct is going to drop into the duodenal and meet the pancreatic duct. So the pancreas is also dropping in its products at the same place, and that is the duodenal ampulla or the duodenal papilla. There's a little sphincter there that's going to help control this and keep things from flowing out, right? So it's pretty much just a little sphincter, and it's going to make sure that you're only releasing bile when you need it, not constantly. So let's take a look at this image, and we're up under the right lobe, okay, of the liver. There we see the gallbladder. Coming down from the right and left parts of the liver, right? Here we have the right and the left hepatic ducts. They join to become the common hepatic duct. That then some of that bile could continue to just go straight out, and that would be the common bile duct. Okay, so we go from common hepatic duct to the common bile duct. The gallbladder here, what I just tell you, that this is kept closed. There's a sphincter here. So as the liver is continuously making bile and that sphincter is closed, it starts to say, you can't leave. So extra bile then kind of gets shunted backwards, and it goes back and it's stored excess in the gallbladder. And that's the cystic duct that is here going up to the gallbladder. At the very end of this thing, we see it dropping in. Again, this would be the duodenum coming into the duodenum, and what we don't see here is that the pancreas is also bringing its substances in here at the same place. This cartoon shows that, although not very well. Here's the pancreas down here, and there's a tube carrying the pancreatic juices, and you see that they're kind of meeting at the same place at the duodenum. Actually, I just messed up there. Let me clean that off. That was my mistake. This is coming down. I was going the wrong way. This is coming down. This is the common bile duct coming down. Here's your pancreas coming in, sorry. And there's your duodenal papilla. Not good when you get upside down. Okay. Yes? Make sense? Okay. So your liver is producing about 1 liter of bile every day. It's a fair amount. It will flow into the gallbladder, and then the CCK will trigger it to be released. So the CCK is going to trigger the sphincter to be opened up, contraction of the gallbladder, and ejection of the bile into the duodenum. So just looking at a mechanism of this. So here's the liver, commons coming, the right and left coming down, common duct coming down to the bile duct, and then here's the backflow, if you will, into and the concentration of it in the gallbladder. As food comes in, especially fats come into the duodenum, CCK is released, and that's going to tell the gallbladder to squeeze and open up this sphincter. And now those fats can be, quote, emulsified, broken down and dissolved into and carried through the digestive system. And then finally we have the pancreas. And the pancreas is, you know where it is. It's sort of behind the stomach. It is largely retroperitoneal, as is the kidneys. It is divided up into lobules as well, and also has 3 parts, the tail, the body, and the head. There is a duct that carries the pancreatic products to the duodenum. That's the pancreatic duct. Here, this is what the pancreas does 99% of. Not 99% important, but 99% of the pancreas is all about making secretions for digestion. Only about 1% of the pancreas by size is related to making hormones like insulin. Okay, so remember those little islets of Langerhans, where the little alpha and beta cells were? Those only account for about 1% of the entire pancreatic structure. So 99% of the pancreas is involved in making exocrine digestive secretions. That pancreatic juice, remember, is quite alkaline. It's helping to neutralize the pH of the chyme. And there's also some enzymes in there that are important for breaking down sugars and proteins. So about a quart of pancreatic juice produced each day, and that's going to then dump into. Now there's some other accessory pancreatic ducts that can occur in some people. They were described by Santorini, so they're mentioned as Santorini ducts, but these are little branches. And why do you think there might be little branches here? The pancreas is doing more than just dealing with fats, right? The pancreas is dealing with a lot of stuff. And so if you only opened up the gallbladder duct in response to fats, then the pancreas would also not be able to drain. So these little accessory ducts are thought to be sort of important in helping the pancreas do its job, even when the sphincter is closed down. If you look at the pancreas under the microscope, it'll look like a salivary gland. It will look like any other exocrine gland with those acinar cells making the enzymes. And again, the islets were those little, quote, islands of cells, the alpha and beta and delta cells, and they were the little endocrine producing cells. So here we see the pancreas. We got the tail way over here, the body, the majority of it. The big head sits sort of right up along the duodenum. You can see the pancreatic duct coming through here, dumping at the duodenal papilla, and you can also see, right, the bile coming down and dumping in at the same place. If you were to look at the pancreas under the microscope, which I won't ask you to do, but what you would see is that the vast majority of the pancreas is acinar cells making secretions, and a small portion of it are the islands of Langerhans that make the hormones. The major enzymes made by the pancreas. The big one's amylase. Amylase. Now, amylase is also made by your saliva. Amylase is the hormone that helps to break down starches in your mouth. When you eat that saltine and it starts to taste sugary, sweet, it's amylase. It's salivary amylase. This is pancreatic amylase. And there's also lipase. So the pancreas is helping break down sugars, and the pancreas is helping to break down fats, lipases. There are also nucleases. We never think about it. We always talk about our diet containing carbs, proteins, and fats. But our diet certainly is also including nucleic acids as we eat anything cellular, right? All things made of cells. We're not eating sand. So as we eat things made of cells, we are consuming DNA and RNA, and those molecules also need to be broken down. So there are pancreatic nucleases that break down the RNA and DNA that we consume. And of course, there are also proteolytic enzymes, ones that break down proteins. So the pancreas is hugely important. Your spit does not break down proteins. Your spit does not break down fats. And your spit does not break down DNA and RNA. So we need the pancreas to finish the job of breaking things down. Now, the pancreas is also releasing a number of hormones that you probably have heard of. Trypsin on the bottom here. Just recognize these as being pancreatic enzymes. Trypsin, chymotrypsin, carboxypeptidase, elastase. These are all enzymes that are released and then activated in the duodenum. So pancreas, very, very important for hormones, but also very, very important, as I'm saying, in digestive system in general. Last little bit, although I didn't mention the teeth per se, I do want you to know a couple of things that can go wrong throughout the digestive system. And one of them, highlighted here, is periodontal disease. This will be a periodontist, a specialized dentist who deals with periodontal disease. This is basically the leading cause of loss of teeth with age. It begins with simple gingivitis, inflammation of the gums. Those bacteria go up further and deeper into the gum tissue and can over time destroy and break down the ligaments that hold the teeth into their place. So when you go to the dentist, the hygienist, they're usually going around and probing the spaces around your teeth, and they're calling out, millimeters, little pockets. And if those pockets get too deep, then you have periodontal disease. The only way to really get rid of those pockets is to go in and what they call scale. So they go down deep in the gum, scale out the bacteria, try to put antibiotics in there to kill those bacteria that are causing the trouble, and hopefully it'll replace itself to some extent. So periodontal disease, you can really see here if you look along the gum line, you can see how receded the gum line is, even below the enamel portion of the tooth, down into the root structures. And so that's a pretty classic picture. And those teeth, as it decays down, become looser and looser and eventually are lost. How do you get it? Not brushing. Not brushing and not flossing. Just not cleaning your teeth well. So when you go to the dentist, they should be checking for pockets, and I'm sure they are. And you really want to go to a hygienist that goes deep, honestly. A little bit of bleeding, a little irritation is okay for a few minutes. You want a dental hygienist who's a little bit more aggressive, I think, who goes down a little bit deeper and cleans well down. And if you walk out of the dental hygienist and you didn't feel a little bit of discomfort, and it was all nice and easy, I'm going to suggest find another one, because I think going a little deeper is better, okay, to get down there and really get that crap out of the gum lines.
So if we ask them to check for that, do we just say are you checking pockets?
They are. They probably are. When you go for your normal dental checkup and you have your cleaning, not so much the cleaning will just be the cleaning, but after the cleaning, right, is usually when the dentist comes in and looks at your teeth having just been cleaned. It's easier now for them to see any kind of caries or any issues with your teeth. And at that point, they should be at least once a year. You'll hear them just say 2, 2, 3, 3, 2, 2, 1, 1, and the hygienist or the assistant is sitting there kind of recording those numbers. They really don't get excited until you're over 5. When you have a pocket of 5, that's periodontal issues. When you get to 10, you've got a pocket there that is significant. Okay. And the problem with those pockets is that bacteria can get down there deeper, right? And they can set up infection. So you want to clean those pockets out, and you want to get rid of those bacteria that can destroy that ligament. So you just kind of go chasing after it. So I'm sure they're doing it, right? It's routine care, but that's what they're looking for. And if you do have a deep pocket, they might send you to a periodontist, and that's a dental specialist who deals with rotting teeth, basically. Another thing about the salivary glands, we don't see this anymore, pretty much because most of us are immunized against mumps. But in the old days, the mumps virus would attack oftentimes the parotid gland, and you'd get this big chipmunk look, and could cause sterility in post-adolescent males. So not only would the parotid gland get attacked by the mumps, but so could the testes. And so boys getting mumps after puberty could lead to sterility, which is one of the reasons, right, that they would get immunized, not only to prevent mumps, but to prevent the possibility of sterility later in life. So most of us now are immunized, and it's no longer as much of a problem. Esophagitis, right? This would be inflammation of the esophagus. Think acid reflux, right? So here you've got a reflux going on. The stomach is percolating too much acid, and the lower esophageal or the cardiac sphincter is not staying tightly closed. That acid is percolating up into the esophagus. The esophagus has no mechanism to neutralize that acid. It's not like the pancreas is spitting out bicarbonate to help neutralize the acid like it does down in the duodenum. So the esophagus is, quote, eroding. And if you're having acid reflux more than a couple times a week, then you are damaging the esophagus, and that can lead to cancer and erosion. So that's an important, in fact, that's become so common, right? There's 3 or 4 drugs now in the aisle for heartburn. Liver, big things here. Hepatitis. There's a couple different viruses, hepatitis A, B, and C. And C is the bad one, right? That's the worst one. Anyone who's working in the hospital will get immunizations against the other hepatitis B especially. The problem is that over time it destroys the liver. The liver can no longer function. It can no longer filter. It becomes scar-like. You're no longer able to fight off, infection as well. So it can be a big problem. If the liver is also not working, you will oftentimes see jaundice. So remember that the liver is helping to break down your blood cells. And your blood cells have lots and lots of heme and hemoglobin, and one of those breakdown products is bilirubin. And if bilirubin is not being properly dealt with by your liver, then your eyes and your skin can become quite yellow in color, being jaundice. So if you look at anyone who has yellowing eyes, there's something going on with their liver. And this is not uncommon either in newborns. So some newborns are born, their liver is not yet fully functioning, and as a result, they have jaundice. And those kids are usually sent home and put under billy lights, right? Sort of a bright bluish neon light. They sleep under that, and what that does is it helps to break down the bilirubin. It's just that the liver is not fully functional at birth. So in those kids, and it's not uncommon at all, in those kids, there's nothing going to be wrong long term. It's just that their liver was not quite up to speed when they were born, and they need that extra help, and that light is able to help break down the bilirubin.
Now, if you're a heavy drinker and you quit drinking, can your liver bounce back if you don't have any of these?
I don't think the liver can regenerate very. I mean, there is the damage to the liver is scar tissue, and scar tissue is not going to regenerate back to becoming liver tissue. It's more of a matter of holding on to what you got, right? So if you damaged a portion of your liver through drug or alcohol abuse or infections, then what you have is what you have. I don't believe there's a way of regenerating. That's why there's so many people on liver transplant lists, right? If the liver could liver could just regenerate, we would not have to do this. But it is true that you can live on portion of a liver. So people have lobes of their liver sometimes. And other thing is gallbladder. We've all heard of gallstones. This is when there's crystals of mineral materials that collect in and no longer can get out, and that leads to a blocking of the ducts, which is incredibly painful from what I understand. Small gallstones will be flushed naturally, and people, when we get to the urine lab in 2 weeks, you'll be examining your own urine, and you'll be, I think, amazed at the crystals that are in your urine. Right? You'll actually be able to see the crystals under the microscope, and those are normal. So you're releasing, epithelial cells in your urine. There are crystals that are made, but when those crystals begin to look like rock candy, right, large crystals, then they can't get out, and that can lead to gallstones. This also can lead to a damaging and an irritation of the gallbladder, and those gallstones can no longer pass. They block the cystic or the common bile duct, and this has to be removed. The surgery has no effect. When you have your gallbladder removed, you're still making bile, right? You're just not able to concentrate it and release it as efficiently as before. So people following a gallbladder removal might complain of the inability to digest a fatty meal, or feeling not feeling well after a fatty meal, simply because the body's not as efficient in dealing with that fat. But life will go on just fine without the gallbladder. There can also be gastritis, inflammation of the stomach itself. Again, if you have ever had irritation from aspirin, alcohol can do this. There can be all sorts of things. If you think about the stomach, you've already got a lot of acid there anyway. So if there's any sort of tear in the lining of the stomach, that can easily lead to gastritis. You can also have ulcers. Ulcers are what? They are a breakdown of the stomach or the duodenal lining. It used to be thought that ulcers were exclusively caused by stress or diet, and now we know that most ulcers are actually caused by bacteria. And the bacteria are treated with antibiotics. And did you come up with that? Did that story come up? Yeah. So the guy who described this was thought to be a lunatic for years. He kept going to meetings about bacteria and about digestive system, and he kept saying, I think there's a link between bacteria in the gut and ulcers. Because at that point, people would drink milk, they would coat the stomach with stuff to kind of coat the lining of the stomach, but it was never thought to be treated with antibiotics. And I forget the guy's name right now, but he actually ingested this bacteria. He was quite sure that this bacteria, this Helicobacter pylori, was the one, right? Pylori. He thought for sure this bacterium was the cause of ulcers, or was a causative agent of ulcers. He actually ingested the bacteria himself, got himself ulcers, and then treated antibiotics and saw the clearance of the ulcers. And so he was able to prove scientifically, and then after that point, people started listening to him. So it was kind of an interesting story where he sort of self-induced this infection in himself to prove that the Helicobacter was, in fact, responsible for about 80% of ulcers. Pancreatitis, inflammation of the pancreas. Again, you got it could be a duct blockage. Some people, when they go in with a gallbladder problem, the pancreas also will be inflamed, and they'll have pancreatitis. The problem here, of course, is that you're not making. If you've got pancreatitis, then you are not going to be breaking things down. Other thing is that injury to pancreatic cells activates lysosomes. And lysosomes can start to break down other things and almost become a positive feedback loop where the lysosomal enzymes themselves destroy the pancreas. So it's almost like an autoimmune type thing. It's not really autoimmune, but the release of lysosome, right? These lysozymes will actually break down the pancreas itself. Enteritis, inflammation of the intestines, that can lead to diarrhea. Dysentery, inflammation of the large and small intestine. Again, you've got bacteria that are involved with this typically. Gastroenteritis, inflammation of the stomach and the intestines. Again, usually involved with an infection, usually from an infection you picked up from dirty water, some sort of disease or bacterium that does that. Colitis would be inflammation of the colon itself. This would either lead to diarrhea or to constipation, either extreme. The diarrhea would be too much fluid. Constipation would be a drying and a lack of fluid in the gut or in the intestines. And finally, we got some cancer that we hear about. Colorectal cancer, one of the more common types of cancer, but now we have great screening. So one of the gifts that all of you will receive when you turn 50 is a visit to the endoscopy, colonoscopy clinic. Right, starting at age 50 now, people usually, as part of their normal routine, get a colonoscopy. If it's clean, if there's no problem, they're usually given a 10-year window. So you don't have to go back and do it for another 10 years. If there's a family history, however, in your family, they may screen you younger and more frequently. So colorectal cancer is very much a genetic cancer. So we know that it runs in families, and so that might be something that would cause you. If you know you have an uncle or aunt or somebody who died of cancer, it really is important that you know what kind of cancer, if they know, and that you communicate that with your doctor, because if it is a colorectal cancer, it's going to change your preventative treatment plan as you move into your older ages. The problem with this is that typically no one is aware of this cancer until it has already spread. So it's slow to develop. These irritations, these benign polyps develop. Those benign polyps then become metastatic. It's sort of like a mole becoming skin cancer. It's a slow progress. And in the colonoscopy, if they don't see anything, then you have a 10-year bill of health. If they see polyps, they're going to send you back more often. And they'll take those polyps just like they would take a mole that was beginning to grow suspiciously. And that brings us to the end of the digestive system. What do you think? Anything of concern, questions at all? Crohn's? Yeah, Crohn's is an autoimmune condition where painful, I've had a few professors actually who had Crohn's, and I've seen people, younger people being diagnosed with Crohn's, some.
I asked because my best friend and her son had it.
Son? Young?
Well, he's now 18,
18,
but he
yeah.
was diagnosed at 14.
Yeah, you'll hear teenagers with Crohn's, and that's not fun.
He
Sometimes
has
it can lead to resection of parts of the stomach and parts of the intestine. Yeah. Okay. One thing I didn't mention in here. What about bypass surgeries? In the classic gastric bypass surgery, do you know what they do?
Take a small piece out.
They staple the stomach into a much smaller section. So really all you have is the cardia, right, the top part of the stomach. And then they will resect the duodenum and bring up a portion of the ileum and connect it to, or jejunum or ileum, and bring it up to the stomach. So in a gastric bypass, what have you done? Not only have you made the stomach smaller, but you've also reduced the absorption of nutrients once it passes into the intestine because you shortened from an 18-foot intestine to much, much shorter. So you have less food going in because of smaller size of stomach, and you have less absorption of nutrients along the way. But what was the one thing that the stomach makes that I told you was necessary for vitamin B12? Do you remember? The stomach makes something called intrinsic factor. An intrinsic factor is necessary for vitamin B12 to be synthesized in your body. So because the stomach has been largely removed, there's less gastric juices, there's less digestion by the stomach. Therefore, what goes through the intestines is not as broken down, less space for it to be absorbed, and that can result in significant weight loss. But it's also possible for them just to keep shoving food down their gut, and it goes right into their intestine rather than to their stomach. So it's not a fix-all, but it is a help. I think that's the last little thing I wanted to mention about the digestive system. Anything else? Our last system for this exam will be the respiratory system. Why don't I give you an early break? Let's take about 5 minutes right now while I reset the clock and get the respiratory presentation ready to go. Okay, so we've been through the digestive system. You're working through the lymphatic system on your own, and that leaves us with the respiratory system. Anyone here going respiratory therapy? Anybody at all? There's usually one or two. I always say at this point, if you are going to respiratory therapy and what I'm about to say bores you, then it's time to change your major. This should be really motivating for those and really exciting for those who are in that program. The first 6 or so modules are pretty much anatomy and review of histology from 105 in lab. This week, you were asked to quickly review the anatomy structures on the models, and I briefly talked about, just in words, what emphysema and what the lung looks like and what the trachea looks like under the microscope. I will go through that in lab next week more carefully, but you definitely want to look through these first 6 or so units, and that's all, quote, anatomy, okay? I will pick up with, and just kind of look through this for major issues. Remember that the respiratory system is broken into 2 major parts. There is the conducting portion and the respiratory portion. And all that means is the conducting portion is the straw. It's everything that brings air down into the respiratory portion. The respiratory portion is where gas exchange actually occurs. The upper respiratory system, there's another way of dividing it. This first division, conducting and respiratory, that's a physiological division, right? It's where nothing is exchanged and then where gases are exchanged. There's also an anatomical division, that is the upper and the lower respiratory system. And the upper respiratory system is everything from the larynx up, okay? So we're talking about oral cavity, and when air is coming into your oral cavity and through your nasal passages, it is being warmed and humidified. You're actually picking up bacteria by the cilia. Those bacteria and viruses are also getting caught up in the mucus being made by the goblet cells, and that's happening all the way through your nasal passages, all the way down the trachea. But the actual upper respiratory system is going to be defined pretty much at the larynx being the divider. The lower respiratory system, again, is moving further down, smaller and smaller structures, and eventually will lead to the capillaries for gas exchange. And I think I misspoke just a moment ago. The lower respiratory is the larynx down, okay? Not larynx up, but larynx down, L. Larynx down is lower, okay, for the lower respiratory system. Now, what is it that's lining your respiratory structures? When I think about respiratory structures, I'm talking about your nasal passages. I'm referring even to your nasal sinuses. Remember the sinuses? Frontal, sphenoidal, ethmoidal, maxillary sinuses, those spaces that are in those bones. Even those are lined by the same respiratory epithelium, pseudostratified, columnar, with cilia and goblet cells, right? Everywhere where air only goes in the upper portion is going to have that pseudostratified ciliated columnar. Now, as you go down into the pharynx, though, what do we find there? What does the pharynx and the oral cavity have? Any place where food would normally travel, will instead be lined by stratified squamous, non-keratinized. Okay. And then when we get all the way down to the alveoli, then we're dealing with simple squamous. So just be thinking about the different epithelia that are lining through the respiratory tract. And this is a beautiful picture of that for your recollection. So again, respiratory mucosa, we're talking about pseudostratified columnar. That is found in your nasal passages and in the sinuses. You've got stratified squamous, oral cavity, and down the esophagus. We're back to the same stuff, right? The same cells are found lining the trachea. As you get down into the bronchioles, that becomes cuboidal. And then eventually, as you get down to the alveoli, you're at simple squamous epithelia. One condition that you have probably heard about, and that is cystic fibrosis. This is a genetic condition. It is considered a lethal condition. People with this condition have a very short life, or relatively short life. It used to be rather short. It has become much longer. Typically, these kids would die before they reach the age of 20. However, now people with CF are living well into their 30s and 40s because of just better treatment. But it is still considered a lethal condition. It is much more common in Caucasians of Northern European ascent, and most individuals with CF, because it is 100% inherited, will choose not to have children to pass this on. Because it was a childhood disease, it's still mostly specialized by pediatric pulmonologists. My PhD mentor had CF and never really talked about it, but every day was going through a lot of treatments to clear those airways. So what happens is that there's a chloride channel blockage, and what that does is that it causes a buildup of the gunk in the lungs. And so they're always hacking up this junk, and that junk also leads to much greater bacterial infections. So they have these almost chronic bacterial infections, chronic hacking of this thick mucus, and not only is the lung defective, so is the pancreas. So the pancreas also is affected by this disease, and in the male, the testes. And so many of these males are themselves sterile because the testes are also affected by this. Not necessarily, and even if they weren't sterile, most would never choose to reproduce simply because of the devastating effects of the disease. But just know that CF, one of those common diseases, relatively common, of the lungs. What I was going to say too is that even though he was in his 40s, he was still going down to the kids' clinic for treatment, right? Because it's the pediatric pulmonologists who are the specialists for CF. So even as adults, they're still routinely going back to see pediatric physicians. Okay, this is just continuing through the structures. We got the nose, we got that. We got the sinuses, right? These are called the paranasal sinuses. Nice little view, kind of comical in a way, looking through those sinuses and looking at this coronal section of the head. Remember that the pharynx is divided into three regions: naso, oro, and laryngopharynx, and that's all described here for you as a review. The nares are simply the opening into nasal vestibule. So vestibule is a generic word meaning opening into. So there's a vaginal vestibule and oral vestibule. Here we see the word nasal vestibule. There was also a vestibule going into the inner ear, isn't there? When you walk past the oval window, you're in the vestibule. Look off to one side, you see the cochlea. Look off to the other side, you see the semicircular canals. The glottis. I know we talked about the epiglottis in the past. The epiglottis is that cartilaginous structure that swings down to block food from not going into the larynx. The glottis, though, right, the epiglottis must be on the glottis. Glottis is referring to the opening to the larynx. So the actual opening is the glottis. Epiglottis is just above it, on it, and that's going to block the opening to the larynx. Okay, so the larynx. I only want you to know the same cartilage that we learned before. So in the larynx, there's a couple big chunks of cartilage. One's the thyroid cartilage. It is the larger of them. It is in males, in some males more prominent. The Adam's apple is part of that thyroid cartilage. And to this are the hyoid bone. Or the hyoid, the floating bone, is also attached here to the thyroid cartilage, as is the epiglottis. The cricoid cartilage is the second largest. It's a little bit more inferior to the thyroid cartilage. And together, thyroid cartilage and cricoid cartilage are really protecting the voice box. They're right over the vocal cord area. So if you take a look at this, you see those two big ones. And again, this is, I hope, nice and reviewish. Big old thyroid cartilage, second largest, the cricoid cartilage, right above the C rings. The glottis, the opening into the vocal folds. So there are two vocal folds. Remember this? There were the true and the false vocal cords. The vestibular folds were just above, and they were helping to basically prevent anything from getting down into the glottis. Because once something gets to the glottis, once you've opened up into the larynx, what can happen? That object can sit on the vocal cords, and if you're lucky enough, you can hack it up and out. But if it gets below the vocal cords, right, it has no place to go but straight down the larynx. Down the trachea. So here you can see the opening of the vocal cords as one is breathing. So here it's showing the glottis is open and the glottis is closed. So as you're breathing, it's the glottis that's opening and closing as the vocal folds open and close over that. To speak requires air to flow past those vocal cords, and just like a guitar string or a piano string or an instrument, like a reed instrument, it's that vibration of those ligaments that's going to cause the sound of your voice. If you have a deeper voice, your cords are thicker. If you have a higher pitched voice, then your cords are thinner. The other qualities of your voice come largely from the size of your sinuses. So those booming radio voices are gifted with larger sinuses that give more resonance to their voice. And if you had very small sinuses, then you would have less of a booming voice. So that's just genetic and just the structure of your own body. Speech is voluntary, do you agree? So all the muscles of phonation, all the muscles of speaking are voluntary muscles. The trachea, you may notice the windpipe has about 15 or 20 C rings that are going to help protect it. Under the microscope, what you'll see again, we'll see it again next week, what you saw this week in lab, is you'll have the C rings made of hyaline cartilage. You'll see those chondrocytes looking back at you, those little round eyeballs looking at you. And then you'll see lining that trachea, pseudostratified columnar epithelium. On the back side of the trachea is the trachealis muscle. The trachealis muscle, if you think of the C rings of the trachea as being like a horseshoe, the trachealis muscle is connecting those two ends of the horseshoe. And just behind the trachea, what does one find? The esophagus, right? So the trachealis is right up against the esophagus as well. You'll see that here in a moment. Remember, the left and right bronchi are different. Which one is larger in diameter, left or right? Right is larger. Why? The right lung is larger, right? Why? Because the heart takes up part of the left side, doesn't it? So the right lung is larger. How many lobes are on the right side? 3. And so therefore the bronchus, the primary bronchus, is also larger in diameter. And that bronchus has more of a direct downward shape to it, because the left side is smaller and has to curve over the top of the heart. The food that drops down, if food were to be aspirated down into your lungs, it would more likely head over to the right side, because the larynx, the trachea heads straight down the right side, and it is bigger in diameter. So for those two reasons, that's where you're more likely going to have pneumonia caused by aspiration. Will be on the right side. So you can see that here in this representation, coming straight down, just a little bit more directly down and bigger in diameter on the right side. On the histology on the right, here's the trachea. This would be the C ring in this area, and the muscle on the back side that would connect the two would be the trachealis here, and then directly behind it you would see the esophagus. And then please tell me what would be right here. Yeah, the vertebral column. Right, so the esophagus is right up against the vertebra. So you've got the trachea, the esophagus, and then pretty much right behind it you would have, your vertebra with the spinous process and everything else. So just make sure you know the flow of air as it travels through both the upper and lower respiratory tracts. The bronchioles. So what is it that keeps the trachea open? The trachea is kept open by the C rings. It never closes, right? Because those C rings are fixed, they can't contract too tightly. But as you go down into the bronchioles, primary, secondary, tertiary, you'll see less and less cartilage. And as you get less and less cartilage, what now keeps those bronchioles open won't be cartilage, but instead will be smooth muscle. And it's that smooth muscle that can become inflamed and close down, constrict down during, for example, an asthma attack. So you'll never, in an asthma attack, the trachea won't close down, but the smaller bronchioles will. So histologically, as you're looking at the trachea, you will see a clear C ring. Histologically, as you go down the bronchioles, you'll see less and less cartilage and more muscle structure, which is responsible for keeping everything open. The parasympathetic and the sympathetic nervous systems are controlling the constriction and dilation of those smooth muscles, much like they do in your blood vessels. So when we think about the bear chasing you, you've got more blood going to the muscles, less to the digestive system. Same thing is true when the bear is chasing you. Your bronchioles will dilate, causing more air to be able to flow through. Bronchioles come in two basic types, terminal and, no, I should say bronchioles, not bronchi. Let me go back. The bronchioles are going to be more and more muscular. And two types here at the end. We've got terminal bronchioles. They're going to be from tertiary on, okay, not the primary and the secondary, but from tertiary on, the third level of branching. And those will eventually go down to the alveoli. And right before you get to the alveoli, there are respiratory bronchioles. And all that means is that in the respiratory bronchioles, there are little side pouches with alveoli. So in the respiratory bronchioles, there is respiration going on there. So if we take a look at this diagram, primary bronchus, and then we see secondary bronchus, and those are sometimes called the lobar. Why would they be called the lobar? Why would the secondary bronchi, the primary bronchi is sometimes called the main bronchi, the secondary bronchi are called the lobar? Different lobes. So how many lobar or secondary bronchi are there on the left side? 2, and on the right side there are 3. So the secondary bronchi are also called the lobar because they go to each of the lobes. And then after that, quite honestly, it's too hard to try to keep track of them in a picture, but they branch and branch and branch. Remember, if you will, if you were in 105 with me, we did the pluck dissection. And if you cut into the lung, if you remember cutting into the lung, it was very elastic, very stretchy, but you would eventually come across these little crunchy bronchioles, rather small, as you travel and cut through the lung at different angles. As you go all the way down, again, what you would have is a nice layer of smooth muscle around that bronchiole. And at the very, very end, right here, is where we have the line between the conducting zone and the respiratory zone. So the conducting zone, everything above that line is where it's just a tube. And then below that line, respiratory bronchioles. So even, if you zoom into this, even in this last little bit of this bronchiole, you'll see a little outpouch. So the little alveoli kind of pouching out of that bronchiole. And those pouches are as effective as any other alveoli. And because gas is occurring there, we're going to call that part of the respiratory portion. As we go down, diameter gets smaller and smaller and smaller, but surface area gets bigger and bigger and bigger. Do you agree? So as we go down, the tubes are getting smaller. As we eventually get down, though, there are many, many, many more of these tubes, and then eventually down to millions of alveoli. So while the tube's getting smaller, the surface area is getting larger. It sounds a lot like your vascular system. As the arteries leaving, smaller and smaller arteries, down to the arterioles. There are more and more and more of these arterioles, and then you get down into the capillaries. So even though you are in a very small vessel, the collective surface area is greater, right? The area that the vascular system is including. So the bronchial tree, we get that idea. There's lobes. There are fissures that separate the parts of the separate the lobes, and then the gross anatomy of the lungs would include a base and an apex. The base would be sitting on the diaphragm. The lung has a hilum. The kidneys have a hilum. We'll see that. The hilum, remember, is where everything goes into or out of an organ. So the hilum would be where the bronchi go in, where the pulmonary arteries and pulmonary veins, all the lymphatic structures, all travel in or out of the lung. So from the backside, you can see this concentrated area where everything goes in and out. That's the hilum. Okay, and then we get down to the little alveoli. And each lung has about 150 million of these alveoli. That means between your lungs, you have about 300 million of them. Recall that every little alveolus has its own little cute little capillary bed surrounding it. But in the capillaries, what are we doing? We're bringing in blue blood and we're sending out red blood. So remember, we're talking about the pulmonary circulation here. So these alveoli are going from blue to red as blood travels through the alveoli and becomes reoxygenated. You already know what the pleura, right? The two layers. And again, as we look at this, don't be caught off guard. Everything you see here in blue is an artery, and everything you see here in red is a vein, right? Because we're in the pulmonary circuit. Each of these little alveoli, you see them here, I'm circling them, has its rich little capillary bed surrounding it. Pretty cool. Now this is new, so this is where I'm going to be picking up and going in slower detail from this point on. So down in the alveoli, there are three types of cells. At this point, when I say alveolus, I hope that you're thinking simple squamous epithelium, and that's true, okay? But we've got a couple of cell types to figure out here. The most common type is the simple squamous cells. Those are called the type 1s, okay? Simple enough. No one was very imaginative when they made these names up. So we got type 1 cells. Those are the most numerous. Those are the simple squamous cells. You might see them called type 1 pneumocytes, right? Cells of the pneumo, of the lung. They're thin. They're all about gas exchange. That's their job. Then there are type 2 cells, or type 2 pneumocytes. These are the cells that secrete surfactant. Surfactant is a detergent-like molecule released by these cells, and what it does is it prevents or reduces surface tension. Okay. Does surface tension mean anything to you? Surface tension. When you fill up a glass of water to the very, very top of the rim, and you notice that the water will even dome over the very top. Why was that? Because water sticks to itself, if you will, through those hydrogen bonds. And you've even seen insects walk on water because of, quote, the surface tension. How tightly held together those water molecules are to one another. What this surfactant does is blows out that ability. Have you ever tried to kill fruit flies in your kitchen? And what do you do for fruit flies? What do you put out to catch fruit flies? apple cider and a drop of apple cider vinegar and a drop of, or cider and a drop of Dawn. Okay, so what the apple cider vinegar is drawing them in, right? It's food. And the moment they touch it, if it were just apple cider vinegar, they could come in and probably sit on top of that liquid and slurp. But you put a little bit of detergent in there, and it breaks the surface tension, and now when they go to it, they fall in and drown. Okay, so it's reducing the surface tension. So that's what this surfactant it's doing. It's acting like detergent, and what that does is it keeps think about it, it keeps the fluid in the lungs very, very, quote, thin, making it easier for gas exchange to occur. It also prevents those little alveoli from closing down. If those little alveoli didn't have surfactant, look at my hand, if they didn't have surfactant, those little tiny spheres would collapse on themselves because the water would be kind of attracted to itself. Okay? So it prevents the alveoli from collapsing. Now, newborns born prematurely do not yet make surfactant, and they're born with respiratory issues because these little type 2 cells are not yet developed. So their little lungs, all their little alveoli tend to collapse. So serious issues, and those kids are usually on respirators as newborn preemies, and they're having to push air in under higher pressure to get air in because of that surface tension that's not being. They actually now will squirt this stuff into their throat, right? So they'll actually squirt surfactant-like material into their lungs, trying to get it to coat so that they don't have to push air in at the same higher pressure, because that can damage these very delicate alveoli. Also, before birth, a mother who is going into early labor will be given some steroid shots. And one of those things that does is it kind of wakes up these type 2 cells prematurely so that the baby will born hopefully with normal lung function or better lung function and save the respiration. There's also down here some roaming alveolar macrophages. These guys are honestly, these are the same cells found in the liver, except they're called Kupffer cells. Okay? In the lung, however, these wandering macrophages are called dust cells. It's the same cell, different place. These same cells in the skin were called Langerhans cells. So we've seen these wandering macrophages by different names. In the brain, they were called microglia. Okay? So we've seen these cells pop up in different places, but in the lung, they're called dust cells. The way I remember that, what are they helping to destroy? The dust, right? So as you breathe in small particulates, bacteria, viruses, and particulates, these macrophages are chewing them up. So 3 kinds of cells down at the alveolus. 2 actually making the alveolus, the type 1 being the most prominent, type 2 squirting out this liquid surfactant, and number 3 are wandering around. These are the dust cells. So here's a cartoon representation of the alveolus. You've got to take a little magic school bus ride with me, and you're down inside. You're looking into an opened up series of alveoli. And imagine, right, this is the alveolus. You're seeing part of it. We see a little opening, a little opening, a little opening. That would be the way by which air comes in to this little alveolus. It is lined mostly by the type 1 cells, the normal little simple squamous cells. The type 2 cells here are shown as these yellow guys. Okay, they're just other cells. They're helping to, they're up along, they're in addition to the type 1 cells. But those are your surfactant secreting cells. And then not attached to, but wandering around are the alveolar macrophages or the dust cells. Now, what else do we see here? This picture is all about the cells of the capillary, of the alveolus. But what else do we see here? It's showing us the capillaries. So let me just point that out to you. I'll use a different color here. This would be the capillary, okay? And this would be a capillary, and this would be a capillary, and this would be a capillary. So you see a dense population of capillaries surrounding these type 1 cells. And it's also pointing out to us that there's a lot of elastic fibers down here. Those elastic fibers are, as the name suggests, giving the lung some stretchiness, some elasticity, so it can open and close repeatedly. So when we get down to the alveolus, I need you to really take an imagination trip here with me. I'll have a picture in a moment. But what is it that an oxygen molecule must do to get into your bloodstream? In other words, as you breathe in oxygen, it's coming down through the conducting zone, all the way down to the respiratory zone of your respiratory system. And imagine now you're an oxygen molecule and you're sitting in this little cave called an alveolus. Your job as an oxygen molecule is to somehow get over to the red blood cells in the capillary, right? It's a very short distance. We're talking microscopic things. But as an oxygen molecule, what layers must you pass through in order to hop a ride on the hemoglobin of the red blood cell? So we've got to get through, number one, the simple squamous of the alveolus, yes? And then we've got to jump into the capillary. And what are capillaries surrounded by? Also simple squamous. So what do we have? We've got a double layer of simple squamous. You with me? And in between that, just a little bit of connective tissue, just enough basement membrane to glue it all together. So collectively, that is referred to as the respiratory membrane. Get these two words straight in your head. Respiratory epithelium, 10, 20, 30 slides back. Respiratory epithelium was what? When I say respiratory epithelium, I'm referring to that pseudostratified columnar stuff with the cilia and the mucus cells, right, and the goblet cells that sit up in the passages of the nasal system. When I'm saying respiratory membrane, I'm referring to this microscopic layer that oxygen and CO2 must pass through. So what is it? The respiratory membrane is the place where gases are exchanging. Very, very thin, rapid diffusion. That means that gases are moving on what property? What's causing them to move? Diffusion is the movement of things going from higher concentration to lower concentration. So what causes oxygen to move in and out of your blood and out of your alveolus is not any kind of active transport, ATP-dependent thing. It's all about concentration. So we've got this very thin, 1/10 of a micron. Okay, 1/10 of a micron. Another way of writing that, that's not right. I'm going blank. One-tenth of a micron. Sorry, I'll come back to it in a second. It'll come up in another place. Senior moment. And so it can be as thin as 0.1 microns and thicker, right? But that's still very, very small, right? Very, very small. 3 layers. We just said them. The alveolar epithelium. We got to get through the simple squamous of the alveolus. Then there's a little bit of a fused basement membrane between them, just a little bit of basement membrane to hold them together. And then finally the epithelium of the capillary. And that we call the endothelium. So this respiratory membrane is right here. So this is collectively respiratory membrane, the 3 layers, right? Alveolar epithelium, connective tissue, and capillary endothelium. So that brings us through the anatomy as well as just a little bit about the anatomy, if you will, of the alveoli, what's going on there. The rest of this is going to be the physiology. Now next week in lab, we'll be doing respiratory physiology. I gave you all a handout. Did everybody get that? Okay. Have this handy. In a few minutes, I hope to get to that place. I should get to this, and I'll describe to you this. And as I describe this to you, you'll also be hearing this again next week in lab. You'll actually be performing this respirometry, or what's called spirometry, next week. And this exact example, ladies and gentlemen, will be on the test. So last week I gave you the blood questions in advance. I showed you the pictures. I told you what questions to be prepared to answer. I'm giving you this in advance so that you are well prepared for this. Okay. Now, what is respiration? When you talk to a cell biologist and you say respiration, what are they first thinking about? What is cellular respiration? Glucose plus water, right? makes CO2 and ATP, right? That idea that every cell takes in nutrients and sugar and oxygen and makes from it CO2 and energy, right? That would be, quote, cellular respiration. In clinical medicine, if you talk about respiration, we're usually referring to the act of breathing, okay? Now, are they connected? Clearly, you have to be able to breathe in oxygen in order to do cellular respiration at each cell. So when we talk about respiration in the clinic, we're talking about air going into your organism as a person. When we're talking about cellular respiration, we're talking about that oxygen now being transported into every single cell, and that oxygen then being necessary to make energy. So from this point on, I'm talking about big time respiration, right? Breathing in and out. So we've got respiration, two parts, external respiration, okay? And this is the exchange of gases between interstitial fluids and the external environment. And then there's pulmonary ventilation. And that would be what we would say is breathing. So that's what I was just describing, these two different types of respiration. What we're usually talking about, though, is the air going in and out of your lungs, that is, in and out of your alveoli. And then, just to be complete, internal respiration, aka cellular respiration. Again, this is referring to the taking up of oxygen into every one of your cells and the release of CO2. Now, what happens? We know that oxygen is picked up in the lungs. We know that CO2 as a gas is dropped off. And I've already told you that that requires diffusion. That means that your oxygen that you breathe in must be higher in oxygen than the blood coming into your alveoli. And that in order for CO2 to leave your blood, the CO2 concentration in your blood must be higher than that of the air in the alveolus. So we'll see this exchange of gases and how this works. Without this gas exchange, there is no respiration of any kind, is there? If you're obviously not breathing in oxygen, then your cells also can't do cellular respiration. So again, just to get an idea of this, hopefully I'm not confusing anyone with this. So external respiration is in and out of your lungs, and internal respiration is cellular respiration, where the gas goes into each and every cell. What if there's not enough air, right? If you are in a place where there's inadequate external respiration, then you would be in a hypoxic situation. Hypoxia, hypoxia, right? So we've got a condition where there's low oxygen levels, and this is obviously going to place a limitation on your cells. So if you don't have enough oxygen coming in, your cells also can't get energy, right? They can't do their thing. There's also anoxia, right? Anoxia would be the complete absence of oxygen. Well, that's going to be death pretty soon for you as an organism. It's also going to be death to your cells. So if you have an anoxic event going to your brain, right, and your brain is not getting oxygen because of a blood clot, right, you're going to have tremendous damage to the localized cells. The organism, right, you may keep on going, but your brain, part of your brain is definitely going to be affected by that. So that's the same also with heart attacks. A part of the heart is destroyed, anoxic, and destroys those cells. Now we're getting, no, I won't say that we're getting into any numbers here, but I will be mentioning a couple of gas laws. We are dealing here with gases, O2 and CO2, and gases behave in unique ways that may have been described to you in middle school or high school physical science classes. And you probably recall that molecules of gas bounce around, right? They bounce around. And the warmer it is, the more they bounce around. The cooler it is, the more they slow down. Gases will bounce around the entire area in which they are found. And there's a relationship between pressure and volume. And this was described by a guy named Boyle. So Boyle's law basically says that pressure and volume are inversely related. This is like taking a balloon. So you've taken a balloon, you've gone down to the store, they've filled it up with air. And what happens? Do you recall what happens if you take that balloon out? Let's say it's winter time, and someone's got a birthday party, and you went and blew up a balloon, and now you bring that balloon outside. What happens to that balloon? Helium or air? Deflates, right? Kind of disappointing. But then when you walk back in the warm room, it blows back up, right? It suddenly repairs itself. It's the same thing with your tires. In the wintertime, what can happen? Pressure goes down. Why? It's colder outside. So what happened? The temperature goes down. So the molecules, yeah. And when it's warmer outside, the gas expands, right? And so we kind of know this. The guy who described this is Boyle's law. Pressure is inversely related to volume. Keep that in mind. That'll be important. Now in this picture, this is showing you the balloon or the tire. So here's the normal situation. Let's say this is room temperature. And if you have, here we've got, you know what, I just messed up. I was just talking about, hold on. I was talking about Boyle's law with temperature, wasn't I? Back up, back up, back up. I messed up, okay? You were jumping out, you were about to grab me, and I was doing my own little mental gymnastics. Boyle's law is pressure and volume. Okay, forget the temperature with the balloon. Okay, that was my mistake. So what we're talking about is simply, if I have a balloon, we are talking about a balloon though. If I take a balloon and I squeeze down on it with my hands, I am doing what? The volume of that balloon is getting smaller. And as the volume of that balloon gets smaller, what's increasing? The pressure. Sorry. Okay, that's Boyle's law. Okay, I apologize. And Boyle's law is about pressure and volume. So here, that's why I said, when I said room temperature here, I was all messed up. So here's your one situation. Now, if I take that balloon, right, as it is, and we assume in Boyle's law that the temperature is constant. So the temperature is not changing, okay? Temperature is a constant. Temperature is not changing. And all I do is I just push in on the sides of this balloon. As it says, decreased volume will increase the pressure. So increase pressure, decrease volume. If instead I could somehow pull out the walls of that balloon, I'd be increasing the, I can't even know what I'm writing here, volume. And I'd be increasing the volume, and that would reduce the pressure. It must be close to the end of the week, right? I am just talking over myself.
Kind of similar, like in the heart, where with the.
This is similar to the heart, right? Perfect analogy. So as the heart is squeezing, the volume is going down, but the pressure is increasing, and that pressure blows open the valve. Perfect analogy. Okay. Perfect. Now, what is happening as you breathe? As you breathe in and out, there's clearly a change in our thoracic cavity. In lab, you were asked to look at the muscles of respiration. Those muscles, the diaphragm, some of your pectoralis muscles, your obliques and intercostals, all those muscles are going to help to expand and to contract your thoracic cavity. And here, that change in volume is going to make changes in the pressure. So that's where we're heading with this Boyle's law idea, that as you breathe, you're changing the pressure and volume of your thoracic cavity. As you breathe, okay, in this situation, this red, we're looking at the diaphragm. And then we have the ribs coming down. And as you breathe, you will see that your diaphragm actually goes down. Okay? So your diaphragm at rest is like a dome. It's an upward dome. It's resting up almost on your heart, if you will. And as you inhale, that diaphragm comes down. And as the diaphragm comes down, what's happening to the volume of the thoracic cavity? It's getting larger. Now, if the volume is getting larger, what is decreasing? The pressure. Okay, I'm going to jump ahead here for a second. As the pressure is going down, air is going to rush into your lungs. And what is causing that air to go into your lungs is the atmospheric pressure. So right now, we are surrounded by an atmospheric pressure that we are so accustomed to that we don't even recognize that it's there. right? We push our arms around in the air and we say, oh, there's nothing there. Actually, there's quite a bit of stuff there, and that's creating an atmospheric pressure. And we're just benign to it. We don't even realize that it's there. So as we increase our thoracic cavity, the volume is going up, the pressure is going down, and now gases will go where there's less pressure. So gases will always go to where there's less pressure. So as you expand your thoracic cavity, air will rush into your lungs. So at rest, we have what's called the intrapulmonary pressure. That's simply the pressure inside, within your lungs. And outside of your lungs, you have the atmospheric pressure. When you're not breathing, right? Everyone just kind of stop breathing for a moment. Just kind of sit there, right? No air is moving. At that point, basically, the pressure in your lungs and the pressure in the atmosphere are equal, right? You kind of, to stop breathing, what are you basically doing? You're intentionally not allowing your muscles to expand or contract, right? So the moment you just consciously stop the muscle changes of your thoracic cavity, at that point, air stops moving in and out. Because at that point, the pressure in the atmosphere and the pressure in your lungs are equal. And where pressure is equal, nothing moves. It's the same as fluid moving through a hose or fluid moving through a artery. What is it that causes the blood to move through your arteries? Pressure, right? Pushed by the ventricles, pushed by the lung, or by the heart. What if there was no pressure? If there's no pressure in a vessel, then the fluid won't move. If there's no difference in pressure, gases won't move. So you've got to have this constant in and out pressure difference occurring in order for air to move in and out of your lungs. So as you expand your thoracic cavity, I've already said this, the thoracic wall will go outward. Remember that your thoracic wall is lined by the pleura, specifically the parietal pleura. That's on the wall of the thoracic cavity. And then there's a space, the serous space or the pleural space, and then there's the visceral pleura, which is shrink wrapped, remember, to the surface of your lungs. As you expand your thoracic cavity, your lungs necessarily are pulled with it because that pleura is like a vacuum seal. So you are opening up your thoracic cavity, your lungs are opening up like an accordion. They're being pulled to the wall. And as that's happening, what's happening to your lungs? As the volume's increasing, the pressure is going down. Again, where will air go? Air will go where the pressure is lower. So as you expand your lungs, more air is going to go in. If you want to take a big old breath, what are you going to do? In order to take in more air, what do you know you need to do? You need to activate more muscles to further expand your thoracic cavity, right? And as you further expand your thoracic cavity, the volume is getting even larger. That means the pressure is going even further down. And as you reduce that pressure, even more air will have the opportunity to be pushed into your lungs. So I know that in an elementary school class, my kids and maybe you were taught that, as you open up your lungs, air is brought in, right, to your lungs. And that's true. But what's really causing it is atmospheric pressure is pushing the air in. And then what happens when you relax those muscles? Your diaphragm then goes back up as a dome, and you relax the muscles. That's causing your thoracic cavity to do what? Get smaller. With smaller volume, pressure goes up. As the pressure goes up, what you're doing is basically forcing the air back out. So your lungs must be working at or about atmospheric pressure. They've got to be bouncing back and forth on either side of atmospheric pressure. If you went to a place with more atmospheric pressure or less atmospheric pressure, you would have a more difficult time breathing or an easier time breathing, right, based upon how your body adjusts to that. So what happens if you were to get a hole in your pleura? This is sometimes called a pneumothorax or a collapsed lung. What would happen? Pneumothorax tells you what? In the thorax you now have pneumo, you have air. If you were to puncture your lung externally and damage the pleura, you've basically broken the vacuum seal. And so now when your thoracic cavity expands and you go to inhale, your lung is not going to, quote, go with it. Now, if your lung can't expand, what cannot happen? If your lung does not expand, that means the volume is not increasing. If the volume does not increase, that means the pressure does not go down. If the pressure does not adequately go down, then the atmosphere can't let that air flow there. So a collapsed lung basically is one that will no longer expand, the pressure will no longer change, and air can no longer get into the lung. So what do we do for that situation? If there is a breakage to the pleura, we just heal it up. We seal it up. We literally plug it, and it will heal itself, because what is the pleura? What is the pleura? What kind of tissue is pleura? Simple squamous, right? It's a mesothelium. It's one of those mesothelial layers. So the peritoneum, the pleura, and the pericardium are just simple squamous, very thin, epithelial. They'll repair themselves, right? And so you plug the hole, let it heal, and usually now, once it heals, the lung will begin to expand once again as normal. Now, this is sometimes the first sign that someone has a lung cancer. Right? If someone has a cancer, the cancer may not have even been visible or noticeable and no respiratory distress has occurred. But if that tumor were to be toward the outside edge and were to grow out and rupture the pleura, that person might have a collapsed lung. Right? So all of a sudden they have a collapsed lung and they go into the emergency room or whatever and they do the x-ray, they see, oh, not only do you have a collapsed lung, you've got a tumor there. Right? So sometimes a collapsed lung is the first sign that there's something more substantial. Other times there's been some sort of trauma, there's been an accident, something else can occur. And sometimes there is an idiopathic collapse, and we don't know why it happens, but it just does. But usually there's a very good reason why the lung collapses. So I'm kind of telling you the story because all I'm going to say to you now is sort of the physical science behind it. Don't be thrown off by the numbers or the, quote, equation. If you understand what I just said, that as your thoracic cavity expands, volume is going up, pressure is going down, and air comes in, that's equated back to Boyle's law, then this will be fine. So at the moment that you have, when you tell, right, when you kind of tell yourself, stop breathing, dummy, right, what happens? To stop breathing means what? You're having to freeze frame the muscles. Hard for me to say stop breathing while I'm talking, right? But if you're not going to have air moving, you have air moving all the time, don't you? Up or down, up or down. The only time it's not moving is when your muscles are at the end of their phase. And then here in this picture, we see this relationship of the pleura and the thoracic wall. So during inhalation, what's happening? During inhalation, the thoracic cavity is going to enlarge. The volume will get bigger. That'll decrease the pressure. So what does that mean? Now the pressure on the outside is greater than the pressure on the inside. Because what did I do? I caused the pressure on the inside to go down. So that difference, right? Don't be thrown off by this, by the math, if you will, the equation. But the outside pressure is greater than the inside pressure. And that will cause air to be moving into the lungs. Turn it around. During exhalation, the thoracic cavity is going to decrease in its volume. That is going to increase the pressure. So now what? The outside pressure is now less than the inside pressure. I'm now pushing the air out, and air is forced out. So that's in words. This is in pictures, okay? So pressure outside greater than pressure inside, air is going to come in. Pressure outside less than pressure inside, air is going to go out. And who's the dead white guy that told us this? Boyle. Okay. There's a couple of the dead white guys I'll be talking about. So that's one of them. Okay, Boyle's the first one. There's going to be Charles in a minute, okay? So all we're talking about then is the movement of pressure, right? Movement in and out. Does that all make sense? Can you picture it? I mean, with a balloon or with your own lungs, you've got to picture this and then let the words settle in. If you could just read anything I put down, you just kind of have to almost imagine it so that you can get this pressure and volume thing figured out. So what is it that happens in Denver? If you were to go to Denver right now, what happens? As Nicole leaves tomorrow on a jet plane, right, and goes to Colorado, what is she going to experience the first couple of days? More difficulty, maybe not in everyday breathing, but if you're exercising, if you go through your normal exercise routine, you're going to find yourself huffing and puffing much faster. What's different? What's different? Not the amount of oxygen, right? That's the elementary answer. It's still 20.9% O2 in the atmosphere. It's the same oxygen. What's different is there's less atmospheric pressure. So what does that mean? That when I expand my lungs here, the pressure drops. The atmospheric pressure is rather significant. The pressure difference is greater, so more air comes in. As I go to Denver and I expand my lungs just as much as I do here in Muskegon, there's less pressure in the atmosphere. Therefore, the difference in the pressure between the outside and the inside is less. Therefore, when I expand my lungs, not as much air comes in. So I will feel as if I'm not getting enough oxygen, right? Now, how does the body respond to that? Remind me. It increases the erythrocytes because the kidneys are going to start producing EPO, erythropoietin. Right? We've been through that story before. But the kidneys are measuring oxygenation. And if they're saying, you know what, there's not enough oxygen coming to the kidneys, they throw out EPO, and that's going to, over time, about 3 to 5 days, right? You're going to feel better in about 3 to 5 days. But the first 3 or 5 days is going to be a little bit tougher. Then when you come back, you're going to be like, ooh, I have a lot of energy. Because your hematocrit will naturally rise, and you'll have more oxygen carrying capacity when you come back. So when you come back, run a race, right? Go play ball. Do something amazing, and you'll be better fit. You'll appear and feel better because of it. Now, this pressure difference here is measured the same way as it is on a stethoscope or on a sphygmomanometer. That is millimeters of mercury. So some of you got caught up on units on the practical. Not many of you, but a few of you, right? Units, 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 units. Always a unit after a number, except on the lab report, right, where there are no units for that area under the curve. But anywhere else, if there's a number, there's going to be a unit after it, okay? No naked numbers on the tests. So these numbers are going to be measured in millimeters of mercury. Graphically, if we just take a look at this, right, inhalation, the pressure is going down. Exhalation, the pressure is going up. That's simply the pressure difference that's allowing you to breathe in and breathe out. Now, what I want to get down to are some measurements, and I know I've only got a couple minutes, if that. I want to jump ahead and introduce this sheet to you. I'm going to skip ahead just a couple slides. And first we got the muscles of respiration. We'll deal with that next time. That's where we'll start. Where I want to go to is, well, it's only about 5 or 6 slides over. Starting right here. This idea of tidal volume, inspiratory reserve volume, expiratory reserve volume. Those are the numbers that you're going to need to understand to do this worksheet and this problem that I gave you. We'll tear this apart in lab next week, okay? We'll tear this apart in lecture next Tuesday. But if you wanted to get a jump start on this, and there'll definitely be some questions about these numbers on the homework, this is where you want to go. Go to these slides. This is 21.10. Here are some equations. It's just adding, nothing fancy. Some addition equations. And where you want to head to is this figure. So where are we? We're in 2110, and you see this graph. Now, the graph that you see there is a teaching graph that looks a lot like the homework page that I passed out to you. So if you want to be proactive and work ahead, this is where this is coming from. If you want to try to figure this out, go ahead and do that. And that's where I'll stop today. So I'll pick up right there with you. Those who have lab next Tuesday are going to be kind of bored with this because you're going to hear this in lecture and lab, but that's okay. Hearing these volumes will be very good to hear them both. And then that leaves us about, 1, 2, 3, 4, 5, 6 or so pages of notes, right? 7 pages of notes. And we'll clearly get through that on Tuesday. So please come next Tuesday. Nicole, have a good time in Colorado. We won't see you next week. But make sure you're looking at your mastering lymphatic quiz. You're doing your digestive homework and trying to work ahead a little bit so that next week you're not panicking too much between Tuesday and Thursday's exam.